people can continue to join while we do the intro.
Sounds
Welcome
good.
everyone to the Washington State Kelp Research and Monitoring Work Group. This is our spring meeting. And as many of you know, we're an open forum for information and sharing and collaboration. We have 3 meetings a year. Our next one is October 18th. And you can get meeting agendas, notes, and recordings on our website. And anyone who either wants to join or leave our work group can just email us at nearshore@dnr.wa.gov. So I'm hoping everyone will introduce themselves in the chat. So if you could just have name and any affiliation or information you want to provide or any updates. And then the other thing is please request topics or let us know of work you're doing or that you'd like to hear about in upcoming work group meetings, because I would hate for us to forget or not hear about some really important work that we want to share as a group. So I'm going to, I do have one quick announcement to make, which is the Washington State Floating Kelp Indicator is complete and online. And I will share a link, I just added it, to the Puget Sound Partnership, Puget Sound info site, where you can see more about that work. Very exciting. Congratulations, everyone. So, and then with that, I'm going to pass it to Elizabeth, who will facilitate our meeting.
All right. Hello, everyone. Up we have the agenda. I think you've already probably seen it in your email. We have rapid fire presentations today. So we'll kick it off with Gray McKenna with Puget Sound Restoration Fund to talk about bull kelp restoration site selection tools for Puget Sound. And then we are just going to move down the list. Each presentation is going to be 15 minutes. Presenters, I'm going to give you a 3-minute warning so that we can stay on track. And you can decide how you want to divvy up presentation versus question and answer. But for everyone who doesn't have a chance to have their questions answered, feel free to put them in the chat and we can try to follow up with you over there and also put them in the meeting summary. So for the sake of time, let's just go ahead and hand it over to Gray, and I will stop screen sharing for you.
Dokes. Can you see my screen, or can you also see my presenter view?
Also presenter view.
Okay, let's try. What about now?
That looks great.
Great. Okay, yeah, good morning. Thanks, Elizabeth. And for folks who don't know me already, I'm Gray McKenna. I use they, them pronouns, and I am the Kelp and Oyster Program Specialist at PSRF through the end of the week. I'm actually about to start a year-long Conservation GIS fellowship with National Audubon Society starting in June, so I'm taking a left turn out of Kelp to do bird stuff for a little while. So this will be my last time participating in this work group as a PSRF rep. Yeah, but I'm excited to talk this morning about a GIS tool I built for Kelp last year as part of the GIS certificate program at UW, which is like a year-long night class program, and myself and a group of 3 other students developed a kelp restoration site selection tool. So I'll tell you a little bit about what we built last year. If folks aren't already familiar with PSRF's work, we design in-water restoration techniques for bull kelp, specifically population enhancement, outplantings, seedings, etc. And we've got this going on at a limited number of sites, but the hope is as these techniques develop and as our understanding of the system develops, we can expand this to more sites throughout Puget Sound. So our project goal was to synthesize available data into a web app that restoration practitioners and resource managers could use to help them with site selection for potential future kelp forest restoration projects. So we created a web mapping application that had two components, an environmental component and a regulatory component, since we need to know, not just where bull kelp grows, but where can we get permits to do infrastructure installations, et cetera. And so for the environmental component, we built a kelp habitat suitability model for Puget Sound, and then we also developed a regulatory reference map. And amongst the student group, I spent a lot more time working on the kelp habitat suitability model, so I'm going to focus on that part for this talk, and I'll show you a little bit of the regulatory layer as well. But for the suitability model, we're looking at the question, where in Puget Sound do environmental and biological conditions potentially support bull kelp growth and reproduction and persistence? And I'll say just off the bat that this is like a huge question, and we had pretty limited time and resources and expertise with which to do that. So for me, this project really represents like an initial stab at this question and what I hope will be the building block for future work. But we're, relatively limited in scope. So I'll just throw that out there at the start. So we began with a kelp literature review, and also I really used the conservation and recovery plan from 2018 as a jumping off point to figure out what input variables would be the most relevant, and then also, of course, what variables we could find publicly available data for. And we settled on this list of suitability parameters for our model, and I'll talk a little bit about each one here. So we utilized the 2014 solution of the Salish Sea model, which is developed by PNNL. That solution is publicly available. We had a temperature layer of mean daily maximum summer surface temperature, a salinity layer of mean daily minimum winter salinity, and then also a mean daily maximum surface current speed layer. For our nutrient and light data, we used the Department of Ecology's Marine Water Monitoring Program, and we had a layer of mean summer surface dissolved inorganic nitrate, and then two different light layers, mean daily maximum summer transmission within 1 meter of the surface, and then mean daily maximum winter light transmission at 10 meters. And for substrate, we utilized the shoreline classification system from 2014, which is, I think, an update of the shore zone data set. And then also the RIGS 1912 historic kelp presence set, the idea there being that if kelp was present and abundant historically, there might be some element of the site that makes it suitable that's not being captured by some of these other parameters. And sorry for the small text on a lot of this. All of this is detailed in a project report that I'll make sure folks have the link to if they want to read more about it. And then lastly, we used a DEM from NOAA to just clip our results to depths that are biologically relevant for bull kelp. So once we had gotten all that data, we did raster interpolations so that everything, looks roughly like what you see here. And then the next step after we get these rasters interpolated is to reclassify them to a common scale. So we used 1 to 9, 1 being the least suitable, 9 being the most suitable. This is just like a pretty classic suitability model scale. And then this was, I think, for me, one of the trickiest parts, or where I felt like we were making a lot of arbitrary decisions because we didn't have sufficient information, where we were trying to define suitability ranges, but there are very few published suitability ranges for these different parameters for bull kelp. So a lot of these are more, the suitability is more along the relationship gradient, right? So hot temperatures in the summer are not suitable, et cetera. And yeah, you can see some of that on the list here. And I don't think I have time to read through each one of them. So I'll just leave that there, and we can circle back if folks want to talk about it more. Then each model input will get assigned the same weight initially. And so each cell in each input is multiplied by this weight and put into the weighted overlay model. And then the output essentially summarizes all of those inputs. And the last step for us was just clipping that model output to 0 to the minus 10 meter mean low low, because of course, even if like the middle of Puget Sound has awesome current speed for kelp, we're not going to find kelp at 100 meters of depth. So the final model output looks like this. And, we went through a process of trying to calibrate the model, trying to adjust those weights to make the model better, more efficient, or whatever at predicting suitability. And the way we did this was to compare the model outputs to the floating kelp distribution data set from the shore zone data set, which is, to my knowledge, the spatial data set for kelp presence in Puget Sound that like stretches that whole region. That the one that I know about that exists. I don't think there's another one that covers the entire Puget Sound yet. Obviously, this is a pretty old data set, but that's what we had to work with. So we extracted all of the cells that fell within that kelp presence and then calculated cell statistics both for those cells within the kelp presence area and for the entire suitability model, and compared the two, and then tweaked our reclassifications and weights to try to maximize the difference between those. So basically what we're saying is that if the model is giving a higher score to those areas with kelp presence in the year 2000, then it's doing a better job predicting suitability overall. Whether or not that's true or the best way to calibrate the model, I'm not sure. But again, this was like a limited time project, so we approached it that way. We built all of it using ArcGIS Pro Model Builder, which is gives you the ability to reproduce the model over and over without needing to do a lot of coding. But so this is how we could iterate through different weighting schemes and classification schemes over and over until we got an output we were happy with. These are the final input weights that we settled with, and I'll just point out that we found that increasing the weight of summer surface temperature and increasing the weight of light transmissions had a big impact on having the model agree more with the floating kelp distribution, whereas the current speed and salinity and the historic kelp presence had less of an impact. Now, there's a lot of limitations to this model. Of course, knowledge of bull kelp biology and ecology in Puget Sound is very much still being developed. So a lot of the model decisions were made based on what we could gather from the literature, but without a ton of great known physiological suitability ranges. And then, of course, we're limited to the data available, which is data sets with different levels of precision and from different time periods. And of course, there may be other variables that we did not have data for that might play a large role in suitability. And because of all that, like I said, we were really placing a heavy emphasis on documentation so that hopefully this can be really a building block, and that as new knowledge and data emerge, people can grab this and run with it and update it and utilize it moving forward. So for the regulatory layers, the thing that we did here was combine a ton of different, jurisdictional layers, information that you would need to complete a JARPA, for example, and did a big layer union so that we had a resulting map where you can click any point and it'll pull up a little pop-up that'll spit out a bunch of information that might be relevant for you if you're thinking about doing any kind of infrastructure installation, which our current restoration techniques do require. So I will see if I can now show you the tool actually. And let me refresh it. Can you see my, this is looking right? You can see the tool.
Yeah, we can see it.
Sweet, thanks, Elizabeth. Yeah, so when you get to the project page, you'll get a little splash screen with some information that everyone will not read and click away from right away. And then you can zoom around and just explore the results, and you can pull up the legend to see what these different scores correspond to and zoom around. You can also have a look at some of the model inputs. Not every single input is on here, but you can look at the visualizations for current speed, for example. You can see that the model score is pretty high for Tacoma Narrows, and of course that corresponds with nice high current speed. And we can visualize temperature, etc. And you'll definitely be able to tell from these that some of these layers have far fewer points from the interpolation, so it's a much like chunkier looking interpolation. Over on the regulatory map, this is one where if you click should pull up a little pop-up that will spit out some relevant information. And there's a couple other layers here as well that you can toggle on to have a look at. And then finally on the About page, you can read some more detailed information and get links to some of these source data sets. And then also there's a link here to our full project report, which if you want to dive in and know everything that we were thinking about and every step that we took, that's all in there. We weren't able to make all of the data like downloadable from this website, as in like our interpolations, but all of that is on hand. And if folks are interested in getting their hands on it, you can reach out to me about that. And let's see, I think I had maybe a few more slides.
And this is your 3-minute warning.
Awesome, thank you. Yeah, so next steps or things that we would have loved to have done had we had the time, I would love to have the tool cover all of Washington waters. Right now, you saw it's just Puget Sound south of Admiralty Inlet. I think there's still a lot of work to be done on improving the reclassification schemes and the weighting schemes as new knowledge of bull kelp emerges. And then something I was really hoping to do, but we could not get our hands on the data fast enough, was integrate future climate scenarios, especially sea surface temperature, into the model so that we're predicting not just where kelp can thrive potentially now, but where it could thrive into the future. And that is all I have. So yeah, thank you very much. And you are welcome to reach out to me. If it's after this week, you should reach out to my personal email, which is graymckenna96@gmail.com. And yeah, that's all I have.
Okay.
So I'm happy to take some questions.
So we have a couple questions. We've got one in the chat, and then feel free if anyone wants to raise their hand as well. So Gray, this is really nicely done. Why did you choose mean winter minimum salinity and mean summer max temp? If you broke this down into winter, spring, summer, do you expect a different result?
Yeah, so the first part of the question, we picked mean winter salinity because I thought that that would be the most likely time that a low salinity event that would be stressful would occur. So that would be, I think, the most likely time that you would see that being a limiting factor potentially for kelp growth and survival and reproduction, etc. And then same logic behind the summer sea surface temperature, right? That high surface temperature when the bull kelp biomass is all at the surface, those hot temperatures that are too hot for kelp are going to occur during the summer. So that's why we selected those time periods. Of course, hot winters are also a problem, and I think, yeah, that would be really interesting to include. And I don't know if that would make a hugely different result, but that would be very interesting and certainly could be pulled from the Salish Sea model and looked at and incorporated as well.
2 quick questions. A request to have the links to the map and report if they're available.
Yeah.
And then another question, do you think there's opportunity to update this via others working on it at the same GIS certificate program?
Yes, I think I've already talked to a few people about this who have asked. I think, yes, there definitely is. Right now, the map and the model is hosted by PSRF, so there need to be, yeah, so you'd reach out to me and then also probably to other folks on PSRF to figure out how that works. But yeah, I think there's definitely opportunity for that, and it's a great program. So yeah.
Excellent. Okay, so we are 1 minute ahead, but I think let's go ahead and stay ahead rather than get behind. You do have some additional questions in the chat, so I just direct you that way. And next we are going to move to Jenny Hahn with Western Washington University to discuss a window into Salish Sea seaweed contaminants and implications for consumption. Jenny, do you have everything you need? You're still muted.
Just doing the screen share here.
Okay.
All right, y'all see it?
Looks great.
Excellent. Well, thank you, Kelp Research and Monitoring Group. I'm honored to be here. And this was my master's work at Western Washington University. So this study was inspired by a question raised by tribes, First Nations, and other community members in seaweed workshops I taught. Are seaweeds safe to eat? We did this research on seaweed contaminant levels and seaweed benefits to answer this question. What was not known are the levels of contaminants in seaweeds and if they are a health issue. And no large-scale study of Salish seaweed contaminants existed. We tested 3 seaweeds, Fucus disticus, Fucus spiralis, and Nereocystis lutkiana. And we did these because these were seaweeds that people were using for foods and medicines. And while we wanted to test sea lettuce as well, we didn't quite have the funds because per sample, it was almost $2,000. So what I know is that most seaweeds are a traditional food for Coast Salish people and that there is a resurgence among tribes and First Nations to use seaweeds. Interest in seaweed farming is increasing, as we all know, in tribe and First Nation territories, and First Nations in the Salish Sea are now cooperating and operating commercial maricultural farms on reserved lands and other areas. Contemporary uses include pickling, wrapping kelp around seafood, soup and stir-fry ingredients, shakers, condiment mixes, and caplets with pulverized seaweed for medicinal use, which you see in the lower right-hand corner. That is fucus that's been pulverized. And benefits, what I also know is seaweed is called a superfood, and it's certainly healthy for you. There are so many benefits. Research shows the beneficial compounds in seaweed may reduce risk for factors for type 2 diabetes, Alzheimer's disease, cardiovascular disease, obesity, high blood pressure, depression, and cancer. Seaweeds have more fiber than most land plants. Rockweed has more calcium than fresh cheese or spinach, and it has twice the iron of beef, and it contains 11% protein. And seaweeds have what are called complete proteins because, like animal products and soybeans and that tiny grain quinoa, seaweeds pack in all 9 amino acids, essential amino acids. However, as you know, using seaweeds from contaminated areas may have health risks. So seaweeds can also uptake pollutants from the environment, store them in their tissues, and potential contaminants in the Salish Sea include metals such as lead, mercury, calcium, and arsenic, and industrial chemicals such as pesticides, PCBs, and dioxins. And while all these contaminants are found across the earth in the water, the soil, the air, and foods, as we know, if their amount is high enough, then they may be harmful to human health. So in this study, we looked for 162 chemicals, including metals, flame retardants, pesticides, PCBs, dioxin and furan, and PAHs, polycyclic aromatic hydrocarbons, which could come from fossil fuels as well as fires. So there are 43 sample sites that I went to, and a couple of the sites are further up off the map near Campbell River. All kinds of boats helped us access the sites. And I do want to say decades before I had kayaked the Inside Passage alone, I ate a lot of fresh sea veggies to lighten my dry food stores. And that kayak came in real handy. But I had a lot of help from First Nations and tribes, biologists, and other volunteers. So we sampled from a variety of beaches, including shipyards, pulp and paper mills, mines, ferry terminals, lighthouses, and beaches where Coast Salish tribes and First Nations have gathered seafood in the past or the present, or where they wish to gather in the future. We also sampled seaweeds from Mussel Watch monitoring sites, which are checked by the Washington Department of Fish and Wildlife. Here are a variety of some of those sites too. So when I typically would go out, I would have permission from a tribe or First Nation to go to the beach, and I went with biologists, sometimes with volunteers, sometimes just myself. This is just an example of the beach at Vashon Island, the Muckleshoot Tribal Beach, and I'm sampling with Andy Dalton and Val Segrist. Andy is the head shellfish biologist, and Val Segrist is a dear friend of mine that's been in many workshops and we work together to share knowledge back and forth. So the lab analysis, we measured these 162 chemicals. We also measured beneficial chemicals via a study that was led by Kathy Van Alstyne at Shannon Point Marine Center, and she was looking at fluorotannins and fucoidin levels. We also worked with 3 labs, NOAA in Seattle and UC Davis, and the SGS Axis up in Sydney, BC. How did we calculate how much bladderwrack seaweed could be eaten on a beach in a week or a day? And this was what our goal was. How could we, in part, say how much you could eat off a particular beach? And that was a question from one of the chiefs that I sat down in a coffee shop and talked with, and it sounded very challenging, but we did figure that out. Once we knew the level of contaminants in the seaweed, we used the approach in making fish consumption guidelines. We used standard calculations with US screening levels established by the EPA, and this allowed us to find out if the chemicals were at a high enough concentration to be of concern for human health. What we assumed in our calculations, since there are no seaweed consumption rate studies for the Salish Sea, and we don't know how much seaweed people are eating, we assumed for our calculations a 175 80 kilogram adult, 175 pound, 80 kilogram adult, that's an EPA standard, and 5 grams of seaweed in dry SDA serving size of seaweed. It's equivalent to about 1 sheet of nori, maybe 2 if it's a thinner sheet. Dave McBride, the toxicologist at the Washington Department of Health, guided this part of our work. So the highlights of the 162 chemicals we measured in Salish Sea seaweed, most chemicals were below the levels of concern. They did not exceed the US-based screening levels. There were 6 chemicals that we wanted to consider further because they were high enough at some sites to be of concern. These are arsenic, cadmium, mercury, lead, benzoapyrene, and PCBs. Concentrations of arsenic, cadmium, lead, mercury exceeded international limits for edible seaweed at some sites. And that's the thing, there are international limits. We don't have limits in our country right now. We're working very hard to establish them through meetings from both coasts. And Europe is also trying to work with some of these standards with the US, so seaweeds can be traded back and forth. For most metals of concern, bull kelp blades were more contaminated than bladderwrack blades and stipes. We also compared levels of 6 of the contaminants, PCBs, benzoapyrene, arsenic, cadmium, lead, and mercury, to their concentrations in a portion of commonly eaten foods, and we found comparable levels. So let's look at one of the sites. This is Freshwater Bay. Let's look where we sampled bladderwrack and bull kelp in 2015. So we made, in particular, Kathy Van Alstyne really led this up, the cards showing beneficial chemicals and chemicals of concern. And again, we focused on the chemicals that we felt were had screening levels and that these ones were high enough to be of concern. So we have here, the top is the green where you could see the beneficial constituents, and the gold below are the contaminants. And we circled in red anything that was above a screening level or of concern. So you can see here that the consumption rate was 3.7 grams dry weight per adult per day, or it was 26 grams, 26 grams a week. And the total PCBs were at this point higher than the screening level. That is what brought this amount from 5 grams per day down to 3.7 grams per day. Just a sec here. All right, so let's look at a way in which I worked out a way to interpret this so it could be understood better, especially by a non-scientific audience, because we really wanted people to be able to look at this and get an idea how much you could eat from a beach based on that one sample from that one day. So at Freshwater Bay, you could take one of these circles that are green, and that is how much you could eat in 1 week. So one form of it would be 5 handfuls of dry tips, and that's about 1/4 cup, or you could eat 1 1/4 cups whole dry tips per week. If you pulverized it, it would be 2½ tablespoons. And if you broke those into teaspoons and people were having this as tea for dietary reasons, it could be 8 mugs of tea. If you were taking caplets, and the caplets were taken for enlarged prostate, for dietary reasons, for low thyroid. So there were a number of health reasons that the caplets are taken. Now, to look at bull kelp, it's a lot more, but you have to keep in mind, bull kelp, we were only able to pay for the analysis of metals. And so in this case, while it says 7 handfuls, it doesn't consider PCBs and other chemicals that would be missing in this analysis, if that makes sense. And I also want to say that the toxicology study was funded for about $90,000. And again, each sample was $1,700. So that's why the bull kelp was not analyzed for more. Let's look at the most polluted site. This is of the 43 sites, Rock Bay. Rock Bay is up in Victoria Harbor. At this time, Rock Bay was under remediation. And if you look over the kayaker's hat, you could see some kind of bales that are golden. That was a whole area set aside with a fencing kind of structure so that when they were pulling out the sediments that were polluted, they weren't getting too much drift. But we were sampling right at that time. I just want to make that apparent.
Jenny, this is your 3-minute warning.
Thank you. And so here we look at bladder rack from the most polluted site. So it's 1.2 grams dry weight per adult per day. And you can see that the total PCBs are almost 3 times as high as the screening level cutoff by the EPA. Benzoate pyrene is very, very high. And this area was very highly industrialized. It was the breadbasket for the Esquimalt, is what the chief had told me. And at this time, of course, 175 years later, it was not a breadbasket anymore and sadly polluted. And lead levels were also quite high. So here you're looking at 1½ handfuls per week and a much reduced level in general. What I want to say too is some people will wonder, so what if I weigh more? What if I weigh less? And this we talked over with Dave McBride at the Department of Health. And he said, if you weigh more, you can eat. If you weigh more, you can eat more proportionately. If you weigh less, you can eat less proportionately. Of course, this does not apply to children, this study. So let's look at PCBs and bladder wrack from the Salish Sea at all the sample sites. So one of the things that we looked at is the cancer slope factor, and that's very much down at the bottom. You can see a line here that is on the lower right-hand side labeled. And up above that, up above that, you can see the RFD at the top where it says 320. This is the standard that fish consumption guidelines are measured at. And so, well, you've got all these bars that are above the cancer slope factor. It's the one above that that is more concerning, or that I should say not more concerning, but is one that is used for the guidelines. Let's compare a portion of seaweed with a portion of common foods. This is something that Dr. Peter Ross had emphasized to me, that you have to keep in mind that all foods are polluted, and many of these traditional foods have so many benefits, not just benefits that are health-wise, but benefits culturally. And so we compared, for instance, here, a portion of bladder wrack to a portion of well-done barbecued steak. And this is all from the Total Diet Study from the US FDA. And you can see here that Rock Bay is still below barbecued steaks and barbecued chicken that's well done. All the other bladder wrack sites are way down here. They're below kale, they're below yogurt. and they're below pumpkin pie, foods that we eat typically, and they're above the hot dog and bacon, for instance. And that is because this food drips fat, it volatilizes, and it spreads the benzoate pyrene on the outside of the food when it's animal fat. Limits of the study, it's one sample from one site. Seaweeds change over the seasons in contaminant and nutrient levels. We looked for 162 chemicals in seaweed. How many more are there? And one of the studies ongoing right now by Dr. Kathy Van Alstyne, Dr. Ruth Sofield, and Holly Souther at Western Washington University is looking at species of arsenic as well as PFA, what do I want to say, flame retardants, PFASs, and they are looking at a few other chemicals as well, including PCBs. So that study is going to be out hopefully in 2024. It's also important to look at a person's whole diet for contaminants, especially other seafoods. And I will say future work is we have a repository for our raw data. We're doing outreach now. And I want to give a big thank you to the 18 tribes and First Nations and my science collaborators at Western Washington University, NOAA, Washington Department of Fish and Wildlife, the CDOC, Washington Department of Health, University of California, Davis as well, and our funders, CDOC and Border Policy Research Institute. So thank you, everybody.
Thank you, Jenny. For sake of time, let's go ahead and direct all questions to the chat. And we are going to move on to our next presenter, which is Robin Fales with UW. And Robin's going to be speaking about understanding kelp sporophyte physiological responses to warming and nutrients across species and populations.
Thank you. I'm working on sharing my screen. How does that look?
Looks good.
Okay, now I just need to organize myself so I have my dual screen. All right, there we go. Thank you for that introduction. Like she said, I'm Robin Fales. I'm a finishing PhD candidate at the University of Washington and Frederick Harper Labs. And I'm going to share with you essentially a snapshot of two pieces of my two chapters of my dissertation. And I just wanted to start by saying that my work has been very influenced by the Puget Sound Kelp Conservation and Recovery Plan, and that I've really the goal to understand and reduce kelp stressors. The timing of when this came out was very influential and really guided the direction of my PhD. And so that's some of the work that I'm going to share with you today. I think this group very much already knows that bull kelp are in decline, but I wanted to point out for Puget Sound from Helen Berry's work that we know that they're the most recent extent. So in pink is where we still have bull kelp, blue is where there used to be bull kelp, and then also here are temperatures and dissolved inorganic nitrogen concentrations for southern Puget Sound. And so in the summer, when temperatures are the warmest, which I've indicated by this dashed line, is also when dissolved inorganic nitrogen availability is the lowest. So these stressors are co-occurring is what I'm trying to get at here. And I just want to acknowledge that we know that bull kelp is in decline in Puget Sound. There's also evidence of declines in Barkley Sound and strong evidence of decline in Northern California being a combination of a marine heat wave and herbivory. So our primary question for this first chapter of work that I'm going to talk about is, does Nereus' thermotolerance differ across the Salish Sea, since we have different thermal environments in different places? And this work was done in collaboration with Helen Berry, Danielle Clare, and Emily Carrington. So we hypothesized that if there was local adaptation or acclimation to warmer temperatures, we would see higher thermal optimums and higher critical thermal maxima. And so to get at what those exact terms mean, here is an example of a thermal performance curve. Here we have temperature on the x-axis and performance on the y-axis. And generally, we expect this trend of a curve shape that's non-normal. And at the ends of this curve, we have critical thermal minima and critical thermal maxima when performance is equal to 0 at that temperature. At the peak of the curve, there's an optimum temperature, T-opt, and then we can think about where the T-opt and the CT max are to inform our predictions about acclimation and adaptation. I'm focused on these upper end of the curve and didn't focus in on the critical thermal minima. We can also take, if we have habitat temperatures for these organisms, we can compare that to these landmarks along this curve and help us understand thermal safety margins and warming tolerances. And so what that is a thermal safety margin is just the difference between our optimum temperature and the habitat temperature. And then the warming tolerance is the difference between the maximum critical temperature and habitat temperature. Okay, so using that framework, I went to a bunch of different sites in the Salish Sea and sampled different bull kelp populations at the beginning of last summer. And I'm just going to give you a quick description on some of these sites. Tongue Point and Turn Rock are both cold sites that are experiencing a lot of oceanic water coming in from the Strait of Juan de Fuca. Cherry Point is a warm site, and so is Squaxin Island, which is the southernmost kelp bed or bull kelp bed in Puget Sound. In more urbanized areas, there's Lincoln Park and Tacoma Narrows, which are both moderate in temperature. And then I also worked with Puget Sound Restoration Fund to sample their restoration site, Jefferson Head, which is seeded from North Beach. So I also sampled that population. And so I want to point out what we know about bull kelp genetic structure from Lily Gerke's work in the Alberto lab. So her work showed that there is there's genetic structure within bull kelp forests in the Salish Sea and other places, and that our cold sites generally group in this Washington coastal category with other open coast sites. From our restoration work, we know that the North Beach population and the restoration site are going to be similar because that's where it was seeded from. And then additionally, Puget Sound groups into another group of differing in genetic structure. So I won't have a ton of time to talk about that, but I think this is really critical for informing how this work relates to physiology. So now, from those different sites that I went to, I built Nereus thermal performance curves. And to do that, I brought my little tissue plugs back to the lab and I incubated them for 36 hours at a normal nitrate level from 9 degrees Celsius to 27 degrees Celsius. And then I performed assays on survival, growth, gross photosynthesis, and panfluorometry. And I'm just going to mainly talk about growth today because this is kind of a snapshot of this information. And so initially, what was really striking is that there was no survival across all sites at 24 degrees Celsius or 27 degrees Celsius. So now, this is a lot of information at once, but here are thermal performance curves for all of these different sites. And so again, temperature is on the x-axis and change in area is on the y-axis as a metric of growth. And then we have our T-opt and our CT max indicated by dashed lines. So I'm going to take this information and show it to you in another way so we can draw comparisons between these different curves a little bit easier. So first, I want to contextualize that with what the actual temperature of the habitat was. So here we have temperature on the x-axis and sites are listed roughly from cool to warm. And so our sites have different mean summer temperatures. And then if we overlay here, now I've added the optimum temperature with 95% confidence intervals that I bootstrapped. And so we can look at what is the relationship between the habitat temperature and the optimum temperature for growth. And you can see that there's a trend where T-opt is increasing with the temperature of the habitat. Now, if we add in our critical thermal maximum, that point when performance goes back down to 0, we can see that that point is higher and it's all around 20 degrees Celsius or 21 degrees Celsius. So across sites, that's very consistent. And so now we can take these comparisons where we have our optimums and our maximums and compare that to the habitat temperature. So essentially I've subtracted those to give us a calculation for how many degrees of warming do we have until we hit a critical threshold. And so for some sites, for the optimum, comparing the optimum for that thermal safety margin, that's the green diamonds, is that we're between 0 and 2 degrees or 3 degrees of warming until we're hitting that precipice where we're going from increasing in performance to a rapid decline. And so that's nearing the threshold for some of these sites, especially at the Jefferson Head restoration. But for our critical thermal maxima, where their ultimate warming tolerance until we hit 0 performance again, we've got a lot more degrees. And so that cold sites, the ones on top, Tongue Point and Turn Rock, have more degrees of warming until they hit that critical threshold. So now I showed you, this is the one on top is what I'd already showed you for growth, but I built these thermal performance curves and did these confidence intervals for all the different assays. And together, I'm just gonna combine this down into a little bit easier to digest information. Together, we can look at across these different assays that we see that there, again, that trend of an increasing with habitat temperature, that there's increasing thermal optima for different metrics. But again, our critical thermal maxima is a 21 to 22 for all sites. So now I'm going to take you down a little bit of a different road and switch gears and talk about a different study. So this work was done in collaboration with Dr. Brooke Weigel, who'll be speaking after this. But for this work, our primary question was, what are the interactive effects of temperature and nutrient stressors on Nereocystis and Saccharina latissima, the sugar kelp? And we hypothesized, because of prior literature on kelp, that there would be higher physiological stress when high temperatures and low nitrogen were combined. So what we did is we used the Ocean Acidification Environmental Laboratory at FHL, which is a fancy laboratory that allows us to control temperature really closely. And we did 2 sets of experiments. In the first experiment, we had 2 different populations of Nereocystis, one from a cold site and one from a warm site. And in the second experiment, from the same site, we collected Nereocystis and Saccharina to compare between species. And for each of these, we had a cool, moderate, and warm temperature, and a high and a low nutrient treatment. And so I'm just going to give you just a quick overview on what these results were. So here we can see, I'm showing you pictures of the blades at the end of the experiment. And the top row is low nitrogen blades, and the bottom row is high nitrogen blades. And then we've got each different column is our different temperatures. And so at 21 degrees and for both experiments, we see decay and decline in condition, but we don't see a lot of difference visually, at least for low and high nitrogen. And so just to give you one of the many things that we measured was growth. And so here we have each panel is a different experiment. In the first experiment, we have Nereo in blue from Turn Rock, and in red from Cherry Point, which is our warm site, and then high and low nutrient treatments. And then in the second panel, we have Nereo from Deadman Island in brown, and then Saccharina from that same site in green. And so generally at cool and moderate temperatures, there's an increase in growth, but across both of our experiments and species, there's a decline in growth at 21 degrees Celsius. But what was really surprising about this work is that in the low nitrogen treatments, our kelp actually grew slightly more than in our high nitrogen treatments, which was not what we expected based on prior literature. Granted, this work was performed over a 9-day experiment, so that's something to keep in mind. But it is very clear from this work that temperature is a strong stressor.
2 minutes, Robin.
Okay, perfect. So now for conclusions for adaptation and temperature. So generally, we found that there was minimal evidence of local adaptation or acclimation other than an increase in T-opt, and that this could be due to a variety of different factors, including connectivity between populations, low genetic diversity, a complex life cycle, which Brooke is going to talk about in just a moment, or seasonality. Additionally, this work, the blades were collected in the field and then had carryover from that condition that they were in the field. And so this gives us a really good idea of what would happen if there was a marine heat waves and temperatures hit these thresholds at that population. But It does, but it wasn't a common garden experiment. Additionally, we found that the max tolerance was 21 to 22 degrees Celsius across these different experiments, and that it was surprisingly the same across populations, and that there's probably been about 3 degrees of increase in thermal tolerance since the 1980s when a similar study was performed from bull kelp on San Juan Island. So importantly, some recommendations for restoration and management is that we need to expand and continue the efforts towards gametophyte banks because we have some populations that are nearing tipping points, and that we should be considering restorative action for populations that are nearing those tipping points of 21 to 22, and we need to be protecting sites that can serve as thermal refugia below the tipping point. If you want to know more about the work that I was talking about, because this is really just a glimpse, yeah, please email me and I'll give you the Zoom link for my defense, which is in 1 week from today. And I'll paste that information in the chat as well. So I'd like to thank my funding and partners. And if we have any time, Elizabeth, let me know if we have some time for questions.
We are at time, so I'm going to throw them in the chat and hand it over to Brooke, who I think is going to build off of what you were just discussing. So next we have Brooke Weigel with UW, speaking about the effects of temperature and nutrients on microscopic stages of the bull kelp.
Awesome. Can everyone hear me all right?
Yep.
Cool. Okay, let me just figure out how to. Okay, sweet. Can you all see my PowerPoint?
Looks good.
Cool. Okay. Yeah. So I am Brooke. My pronouns are she, her, and I'll be talking a bit about this project that I did over the last 2 years as a postdoc at Friday Harbor Labs. So today I'm focusing on bull kelp, Nereocystis leucana, as we've heard from the last few presentations. And I'd like to acknowledge the work of Sadie Small, who was my research assistant on this project last summer at Friday Harbor Labs, as well as Naomi, who was our RU student last summer, and she is super awesome and hopefully will continue to be part of the kelp world. So we know that there's a lot of complex factors relating to the loss of kelp forest ecosystems, and we see really different regional differences in stressors. There's some areas where we have loss of Pycnopodia and an increase in urchins, like in California. But here in the Salish Sea, we think that thermal stress combined with other factors are really important because we're losing bull kelp in areas where we don't see a lot of urchins. So we are seeing substantial bull kelp forest declines in Central and South Puget Sound. And as Robin just mentioned, a lot of the areas where we're losing bull kelp get really warm in the summer. And at the same time, the water gets stratified. So we have low dissolved inorganic nitrogen concentrations in that warm stratified water. And so I'm really interested in looking at the interactive effects of these two stressors because they occur simultaneously in the field. And in order to separate the effects of high temperature from those of low nutrients, you really have to tease apart these two factors with lab experiments. And further, I think it's really important to understand how environmental stressors impact bull kelp across the whole life cycle, because obviously we need kelp to accomplish all of the stages of their life cycle in order to persist year after year. And this is especially important for bull kelp because it's an annual species. And so we have the adult sporophyte, which releases these microscopic zoospores that turn into gametophytes and juvenile sporophytes, which are living at the bottom of the ocean. And Jordan Hollersmith did this really nice review of stressors for kelp forests in the Salish Sea. And I just want to highlight that in these pink, orange, and green colors, there's very few studies on all kelp for their microscopic stages with respect to both temperature and nutrients. So just to refresh the bull kelp life cycle, for those of you who might not be familiar with it, we have the adult kelp sporophyte, which grows up from the early spring throughout the summer. And sometime in early to mid-summer, they form these reproductive sori patches on their blades. These sori patches release microscopic zoospores, which settle to the bottom of the ocean and germinate into separate male and female gametophytes. The male gametophyte then releases sperm and fertilizes the female gametophyte, and then that grows into the juvenile sporophyte. And so the project I'm talking about today focuses on the effects of environmental stressors on these microscopic stages that are usually living at the bottom of the ocean. So in this study, we sampled bull kelp from 7 different populations, starting with Gordon Island in the North San Juans, Turn Rock near Friday Harbor Labs, and then down into Puget Sound, we had a population at Edmonds, Lincoln Park, East Vashon Island, Tacoma Narrows, and then Squaxin Island. And at each population, we collected sori patches from 15 different individuals and brought them back into the lab at Friday Harbor and released the microscopic zoospores so that we could grow gametophytes in the lab. And these gametophytes were grown across 7 different temperatures. So we had temperatures ranging from 10 to 22 degrees Celsius in order to capture the whole wide range of environmental temperatures that they might experience, particularly the warmer end. And then in addition, they were grown at, within these 7 temperatures, they were also grown at 2 different nutrient levels. We had low nitrogen, which was just ambient seawater, and then high nitrogen, which we spiked with an additional 30 micromolars of nitrate. And I took nutrient samples showing that the nitrate was depleted in the low end treatment after a week or so, but it was still remained high in the high end treatment. Okay, so I'm going to show you a series of graphs. They all look like this. So let me walk you through these graphs. On the y-axis, we have our response variable, which in this case is the growth rate of the gametophytes in millimeters squared per day. And on the x-axis, we have temperature. And then each separate graph is a different population going from south to north. And then the points will represent the different nutrient levels. So blue is high N and pink is low N. So we found that gametophyte growth rates peaked at about 16 to 18 degrees across populations. And there were significant effects of temperature and population, but notably there was no significant effect of nitrogen on gametophyte growth rates. Importantly, across every single population, gametophytes died at 20 and 22 degrees Celsius. So you can see here there was no growth at those 2 warmest temperatures. Next, this is the density of gametophytes in each culture. So you can see that the gametophyte densities peaked at colder temperatures like 10 to 14 degrees Celsius. The densities at 18 degrees were 95% lower than the densities at colder temperatures. So by 18 degrees, the densities declined significantly. And then again, the gametophytes were dead at 20 and 22 degrees. Again, you can see there was no trend in nitrogen for gametophyte densities, but they were really significantly impacted by temperature and showed pretty much the same trend across all populations. So we grew this experiment until they turned into juvenile sporophytes, which was started happening after about 2 weeks, but we let them grow for, I think, 40 to 60 days and looked at the response on juvenile sporophytes. So pictures are pretty clear here. You can see that sporophyte production peaked at colder temperatures. There were a few sporophytes at 16 degrees, like 1 or 2 at 18 degrees, and nothing at 20 and 22. So I'll show you that quantitatively. You can see here, this is the density of those juvenile sporophytes, and sporophyte production peaked at 10 to 14 degrees Celsius. there was a slight effect of nitrogen, and you can see for some populations, the blue points are higher, so there were slightly more sporophytes with additional nitrogen, but only at cold temperatures, because again, there were no sporophytes at 20 and 22 degrees, and a lot of populations also had no sporophytes at 18 degrees, and the sporophyte production at 16 degrees was super limited. So sporophyte production was really limited above 16 degrees across all populations. And then finally, we measured in image J the size of the sporophytes, and sporophytes were also larger at colder temperatures from 10 to 14 degrees. And this is the only metric where we did see a more significant effect of nitrogen. You can see that the blue dots for some populations were quite a bit higher, so they were larger with more nitrogen. But importantly, additional nitrogen did not improve sporophyte survival at high temperatures. So it can give them a bit of a boost at cold temperatures, but regardless of nitrogen levels, they did not produce sporophytes at these high temperatures. So my research demonstrates that there's different thermal performance curves across the life cycle of these microscopic kelp. The gametophytes seem to have a bit of a higher optimum for growth of 16 to 18 degrees, while the sporophytes really performed best at colder temperatures of 10, 12, and 14 degrees. So in order to sort of ground truth these results and see how warm it's actually getting at the bottom where the gametophytes are growing, I went out and did a bunch of scuba diving in collaboration with a lot of others and monitored subtidal temperatures across these sites. And thanks so much to all the divers and snorkelers who helped with this. Many of them are on this Zoom call today. So here's a graph showing daily mean bottom temperatures. So these sensors were all placed at -3 to 5 meters relative to mean low water in kelp forests. And you can see that daily mean temperatures were generally below 16, but at Cherry Point, daily mean temperatures did get above 16 degrees for 6 days. But when we look at the daily maximum temperatures, there were 4 sites that exceeded 16 degrees on many days, including Squaxin Island, which sat above 16 degrees daily max temp for 54 days, pretty much the whole summer. And then we also see a few sites like Squaxin Island and Cherry Point that exceeded 18 degrees. So it is getting really warm on the bottom at these sites. So my research has shown that 16 degrees is sort of a critical threshold for production of those juvenile sporophytes, while the gametophytes are a bit hardier and were able to grow up to 18 degrees. And then my collaborative research with Robin Fales, which Robin just talked about, showed that 21 degrees is really a critical temperature for the adult sporophyte blades. So it's really important to continue monitoring temperatures throughout the Salish Sea because they clearly have a really strong impact on bull kelp across the life cycle. And I think the next step is really digging into understanding the phenology of this kelp growth and production of these juvenile sporophytes in the field relative to the period at which we have these warming temperatures. And I'll note that my study was sort of like the worst case scenario. We held them at these hot temperatures for, a month and a half straight. And realistically, they're getting shorter heat waves of 5 to 10 days of these warm temperatures. Although at Squaxin Island, they were hitting 16 degrees every day for 15 days. So this scenario isn't entirely unrealistic. And with that, I'd like to acknowledge the Washington State Legislature for funding for my postdoc. I had a really great time, thanks to my advisor, Megan, and again, Sadie and Robin and Naomi and everyone at DNR, as well as Friday Harbor Labs. If you want to reach me, I'm currently a postdoc working at Western Washington University looking at the effects of climate change on alpine snow algae. So I switched from the giant algae to the microscopic weird guys that live in the snow, but I'm still around Washington and still happy to be involved in the kelp world. And with that, I'll take any questions.
We maybe have time for one question. I see Hugo has asked, what produced the changes in temperature?
You mean in the environment, what produced it, or in the lab?
Hugo?
Sorry. Yes. I'm interested in knowing what it seems like the temperature is a huge stressor. And if we don't have a lot of information on that, do you have any idea of what created those changes in the temperature and the
You mean
environment?
in the
Yes.
environment?
Yes.
I mean, I think it has to do a lot with like what Gray was showing with their modeling. There's just sites that have really high current that get a lot of flushing of fresh ocean water, and sites that have low current and low turnover. And I think the depth also matters a lot. But I mean, we see the warmest temperatures at places like Squaxin Island, which doesn't get a lot of flow and is furthest south. And then we also see warm temperatures up at Cherry Point, which are probably influenced by the outflow of the Fraser River. So I think it's pretty complex.
All right. Thank you.
Yeah.
Okay, thank you, Brooke. I think I'm going to direct all other questions to the chat for you.
Awesome. Yeah, please ask more questions. Also, we wrote this into a paper and it's in review right now. So hopefully I can share it all with you open access pretty soon.
Great, thank you. Okay, we are going to hand it to Tim McClure at DNR to talk about a tale of two kelp beds in South Puget Sound. Thermal stress in 2022 accompanied loss of canopy and understory kelp at Squaxin Island, while Salmon Beach remained stable. So kind of following up on what Brooke was just speaking about. Tim, your slides look good.
Perfect. Can everyone hear me okay?
Yep.
Perfect. Okay. So thanks for the introduction. Yeah, my name's Tim McClure. I use he, him pronouns, and I work in the nearshore habitat program in the Washington Department of Natural Resources. Today, I'll be telling you about our kelp monitoring work here in South Puget Sound, which really kind of is a tale of two kelp beds. So the first thing you notice when you jump into the history of kelp in South Puget Sound is that we have been having declines on actually a really broad time scale. What I'm showing you here is a map of South Puget Sound. Robin also showed us earlier with the areas that had kelp historically in blue and the areas that have kelp more recently in pink within the last decade. What you'll notice here is that Squaxin Island really stands alone amidst a lot of blue, and it's actually been the innermost kelp bed in Puget Sound over this duration. And overall, in the last 145 years, over 80% of the bull kelp has disappeared from the shorelines in South Puget Sound. And so what our monitoring work in the last decade has found is a nearly region-wide decline, with many sites experiencing total losses. And so zooming in now on Squaxin Island, what our monitoring has shown more recently is two distinct periods of canopy decline at Squaxin in the last decade. What you can see here on the right is a map of its kelp bed area, where the purple color tones are the older years, and you can see how the bed area has shrunk both along shore, parallel to shore, as well as perpendicularly, the outer deep edge of the kelp retreating into shallower water. From these estimates of bed area, Squaxin has decreased by about 97% in the last decade. And if you look at a graph of these over time, the distinct periods of decline become more apparent in 2013, 2014, and again, more recently in 2021. And here the thermometers indicate the coinciding timing of regional marine heat wave events. And so this is in contrast to the bed at Salmon Beach, which is near the Tacoma Narrows, shown here using the same color scheme as Squaxin Island. And here and there we can see some interannual variation at the outer edge since 2018, but overall, the bed has not shrunk along shore or into shallower depths. And so these differences raise natural questions regarding the mechanisms responsible for these declines, as we've been hearing about so far today. And in recent years, ongoing research has been occurring into identifying and understanding these stressors associated with the declines. A major recent finding from our collaborators at Friday Harbor Labs, who we've just been hearing from, is that thermal physiology limitations occur at field relevant temperatures here in Puget Sound. Brooke and Robin have conveniently already covered a bunch of the detail in their work and also the life history cycle for kelps. And so just as a refresher, they found that gametophyte densities were highest at 16 Celsius, but declined starting at 18 Celsius. And additionally, the gametophyte growth rates were highest at 18 degrees, but had zero growth and high mortality beginning at 20 degrees. And additionally, they found that the sporophyte production peaked around 14 Celsius and was declining once temperatures reached around 16 Celsius. And so for the, There we go. And so for the purposes of these results I'm showing you today, the big takeaway from this work is that temperatures between 16 and 18 degrees represent some concern for bull kelp in the region. And so with this in mind, in 2021, the Nearshore Habitat Program began deploying arrays at these two sites to assess any abiotic changes in conjunction with these canopy declines. And additionally, the GUI Duck compliance divers completed biodiversity surveys at both these sites in 2021 and 2022. For the sake of time today, I'm going to skip some of the details surrounding the exact protocols used for these dive surveys, but I'm happy to answer those types of questions later, and also keep an eye out for our report on this work coming soon. With that said, in 2021, the dive survey used the protocol originally developed by PSCO, the USGS, and the Olympic Coast National Marine Sanctuary, with which Puget Sound Restoration Fund has been conducting for several years. And in 2022, the dive surveys used the Reef Check Washington protocol. A huge shout out to our partners, both at Puget Sound Restoration Fund and Reef Check, both of them on the call today. As you're about to see, these surveys contributed new insight to the ongoing dynamics of these sites. For the purposes of today, though, what's important to understand is that these surveys completed a series of fish, algae, and invertebrate surveys, as well as a substrate survey at both these sites in a layout that kind of looks like the diagram here. And so this gets us to our research questions on my project, which we've observed elevated temperatures at Squaxin Island, but what was the frequency, intensity, and duration of these temperatures? And secondly, how does the temperature at Squaxin Island in particular compare to these concerning temperature ranges for bull kelp? Secondly, what's happening below the surface? More specifically, are there similar patterns or shifts in the understory community occurring? Or were there any shifts in the site characteristics that could have affected the kelp habitat suitability? And so first, jumping into our temperature results. What I'm showing you here is a heat map of the temperature measured every 15 minutes at both sites. Again, the sensors that Brooke was talking about a minute ago, with Salmon Beach in the top row and Squaxin Island in the bottom. Time runs left to right and top to bottom in these figures, where the x-axis represents sequential months, and then within each block of month represents the day of the month, and then time of day runs from top to bottom. And in this figure, the warmer temperatures are indicated with the red color tones. And so zooming in on Squaxin Island, what we see is that Squaxin experienced distinctly higher temperatures in the summer months than Salmon Beach. And additionally, this heat map really distinctly showed us that the temperature extremes occurred around midday and also weakly coincided with some patterns in tidal timing. And so spending some time looking at these heat maps got us thinking about the durations of exposure to these concerning temperatures, but also within a day in the field. And so what I'm going to show you next is a table figure of the temperatures between July and September in both years at Squaxin Island, where the longer durations above 16 degrees Celsius in a day are shown in red on the left. And here on the right is a similar graph displaying the durations where the tidal levels were below 0.3 meters or 1 foot above mean low or low water. And so darker shades of blue on the right indicate the more extreme low tide events. And what we found is that there were durations up to 17 hours with hourly mean temperatures above 16 degrees Celsius in the summer months at Squaxin Island. But more notably, we also found that the longest durations of highest temperatures actually didn't coincide with the longest low tides as I expected, but rather was actually offset by about 5 to 7 days. And so this was actually occurring during the neap tide weeks and these periods of lower tidal exchange and currents. And additionally, this trend was a bit more prominent in 2021 than in 2022. So lastly, when looking at the data summarized by day, we found that the daily maximum and mean temperatures at Squaxin Island were at the range of concern that we have for bull kelp. First, what I'm showing you here on the left is similar plots again to what Brooke just showed us, where the daily maximum temperature is on top. 16 degrees Celsius is indicated by the dotted line, 18 degrees Celsius indicated by the dashed line in both figures. And again, the top is the maximum temperature and the bottom is the daily mean temperature. And we found that the daily maximum temperature at Squaxin exceeded 18 in both summers in 3 specific instances, but actually otherwise remained below this threshold and was regularly exceeding 16 Celsius. And as Brooke pointed out earlier, this was for a total of 54 days in the summer. And so secondly, daily mean temperatures reached 16 degrees Celsius in both summers. And again, this is a temperature that's associated with these declining sporophyte production and thermal performances in the mesocosm experiences we were hearing about a bit earlier. And so now to jump into the results from the dive surveys that we had done, what we found was that declines in the understory kelp community also occurred alongside our observation of bull kelp declines. And so what I'm showing you here on the right is a plot of the understory densities separated by the different species on the left, with magenta and pink colors representing kelp species and blues representing the other algae species in the survey, and then summarized here on the right, likewise by kelp shown in darker gray and non-kelp in the lighter gray on the right. And what we found was that in 2021, the understories were dominated by sugar kelp, Saccharina latissima, which again we heard a bit about earlier. But in 2022, what we found was that Squaxin Island experienced a 92% decline in kelps and had partial replacement by acid weed. And then this resulted in a 51% decline in overall understory density. You can see that here in the figure, looking in the left column for Squaxin Island, really this big transitional shift from magentas to blues, and then the overall decline here on the right. And again, again, this was driven by these increases in acid weed. And so the second piece of the puzzle in this system that the dive surveys allowed us to start thinking about was whether or not there were other changes in the community, such as habitat or substrate composition, that might have affected the kelp abundance between the years. So what I'm showing you here on the right is another similar stack bar chart, but with percent composition for each substrate category. And yellow and tan color tones represent these smaller substrate types, like sand, shell hash, and pebbles. And the darker grays and blues represent the larger types like boulders and bedrock reef. And what we found was that Squaxin Island does have more sand and smaller substrates overall than at Salmon Beach, but these percent compositions did not change substantially between years, which means that changes in the habitat suitability are unlikely to be driving the observed changes in the kelp density. And so that really brings us to one of our main conclusions in this work, which is that pairing underwater surveys with the environmental monitoring efforts contributes new and also unique insights into the interactive effects of stressors. We found that the temperatures at Squaxin Island are starting to reach these ranges associated with physiological damage in the summer months, and that these physiological damage limitations represent a major concern for continued warming in South Sound, as well as the region as a whole. And additionally, we found major decreases in density and community shifts in the understory alongside these declines in the bull kelp canopy our existing work was identifying. This also highlights some additional mechanistic details, including the role of competition in these understory community shifts, and as well as the importance of continued research into other interactive effects of stressors, like nitrogen availability, which again, this is the broader topic that Robin and Brooke covered so effectively a minute ago. And lastly, this decline also reflects the potential for lower recruitment in subsequent years following these warm thermal events. And so putting all this work together really highlights the continued importance of more broad understanding in the system, the type of work we've been hearing about for the last half hour or so, also occurring in the understory, for example, what's happening in the juvenile life phases of understory kelps like Saccharina. These are all questions for the future for us to explore. And so with that, I hope that I've left a couple minutes for questions. I know that was a lot, and I'm sure there's some specific questions. So with that, I'd like to switch to answering any of those.
Thanks, Tim. I think we have time for one or two. So I don't see anything in the chat yet. Anything, feel free to use your hand if you'd like.
This is Megan.
Yeah.
I have a question.
Go ahead.
So Tim, did the Desmarestia just show up early in the spring and then disappear, or was it there making trouble for much of the season?
That's a fantastic question that the data doesn't directly answer for us. These surveys were completed in late July and in early August, so we don't have specific understanding, at least from that data, in terms of the phenology and the timing of some of these different species. with that said, several of our staff are out there, and I'm sure Helen could chime in with some personal observations. But based on the diving I'm doing this spring, it seems like the acid weed does kick in a little earlier. So Helen, if you've got anything to add, that'd be great as well.
I was just typing madly. We do see the Desmarestia continue until late in the season, whereas we really see the Sargassum die back. So very different between those two genera.
Okay, we're on it.
I see Tom's hand next. I don't know if we have more time, Elizabeth, but.
Tom, if you're quick, we have time.
I was going to just add a little bit to that, asking about the interactions with the Sargassum as opposed to Desmarestia there. Is what do you think that Desmarestia is a bigger problem?
I think from this data, what this data can show us is that in terms of the distinction between kelp species and then non-kelps in the overall understory community, that there is for sure a transition away from these kelps. I don't think we can opine from these data on which is more of a problem, but I think the patterns in the phenology and the associated patterns in temperature mean they're both concerns, for sure.
Let's continue to talk about palatability and who eats who with kelp crabs here. That might be interesting to follow up.
100%. Yep.
Okay, that's probably time. Thank you, Tim.
Cool. And I'll keep answering in the chat. I see some other questions. Thanks, everyone.
Okay. And Brian is on it. So we are going to move to Brian Timmer with University of Victoria. And he's going to be covering a better understanding of remote sensing and historical ecology of kelp forests in the North Salish Sea. I will just hand it to you, Brian.
Hello, everybody. Thanks for having me. Good to be here. I'm realizing I probably should have just scheduled 2 talks or something like that to fit this all in, but we're going to fly through some stuff. So first, I'm going to chat a bit about my master's work, which is also here at UVic in the geography department with Dr. Maisira Costa. And that was looking at the effects of submersion with tides and currents on the remote sensing of surface canopy forming kelps. Was done in close collaboration with the Hackeye Institute and all of the wonderful groups you see on the screen here. Sorry, my computer is freezing. Oh, there we go. Cool. So basically the goal of my master's was, just to understand better how the submersion of these kelp canopies affects our ability to actually detect them with remote sensing. And so we can kind of think about this in 2 different ways. One of them is from the structural level, thinking of the actual individuals themselves, and one is at the bed level, thinking of how on a broader scale these kelps are being submerged. And so just a bit of a background for those of you who may not be super into the remote sensing world. Basically, when we're thinking of kelp remote sensing, this is basically talking about with multispectral. So seeing into the wavelengths of light past the human range. So we as humans generally see about 400 to 700 nanometers. And just beyond that, you get into this area called the near infrared. And so on this plot on the left, you can see there's reflectance on the y-axis, and that's basically light bouncing off of any object and coming out and going into your eyes, and that's what you're actually seeing is the reflected light bouncing off of something. And then on the x-axis, you see the wavelengths of light, and so beyond 700 is where you see the near infrared signal of kelp. That red line gets really high, and that of water stays quite low. And so that's what we can use generally with satellites and drones to determine whether or not we're looking at kelp. And so on the right side here, you can see an example of how this works. Basically at a low tide at 0.3 meters, there's lots of kelp on the surface. And as you go up, the kelp is submerged and you lose that strong near infrared signal. And so we actually did some experiments where we submerged kelp and took hyperspectral data. So this is like continuous wavelengths of light along the x-axis on the bottom here. And again, reflectance of light on the y-axis. And you can see in the inset plots that at the surface, the Nereocystis kelp bulb and blades had this classic high near infrared signal. But once we submerged it, you ended up getting this interesting phenomenon where around 700 nanometers, where we consider the red edge, sort of the lower side of that near infrared, you had this peak that was always higher than the higher near infrared wavelengths. And so based on this, we often will be using those like around 800 or so nanometers to actually detect kelp. And so this was showing that in pretty much all cases where kelp was submerged, the red edge actually gave a stronger signal. And then when we looked at in situ, like how the kelp actually sits at the water surface, we found that there were some differences between bull kelp versus giant kelp, and even differences between bull kelp bulb and blades. So with the bulbs, these large structures floating at the surface, they're actually emergent above the surface of the water. And so you get that classic high near infrared signal, with the near infrared being actually a little bit higher than the red edge. But once we started looking at macrocystis fronds, where the blades are largely below the surface of the water, and nereocystis blades, where they're also below the surface, you started seeing overall a lower magnitude of signal, so basically just harder to detect, as well as, again, higher signal in the red edge versus the near infrared. So the reason for this was basically that the water, the red line here is showing how water will attenuate at these wavelengths of light. So going up and higher into the near infrared, you get this exponential curve basically of attenuation. And there's this little sliver in the red edge region that the light can actually penetrate past that high attenuation. And that explains why we can actually see a little bit deeper into the water. So all of this is interesting from a hyperspectral remote sensing point, but for practical purposes, usually if we're doing drone or satellite mapping, we have this multispectral data instead with very discrete bands of light. And we'll often calculate things like NDVI, the normalized difference vegetation index, different indices to just make it nice and simple to classify kelp as sort of yes or no, it's there or it's not. And so we simulated this data into multispectral bandwidths, and we did a whole bunch of different vegetation indices. And so this is a bit of a complicated graph, but very simply put, what we did is we took different vegetation indices, so different combinations of bands. And on the top row here, we have red edge combined with blue, green, and red. And on the bottom row, we have the near infrared band combined with blue, green, and red. And basically, what you're looking at is as these dots on the x-axis, you can see that that's associated with depth. So as these dots cross this red line within each of these plots, that means you can no longer generally detect the kelp. And so basically what this is showing is that with the red edge indices, in all these cases, you can basically see kelp down to about a meter deep, whereas with the near infrared by 30, 40 centimeters, you're losing your ability to detect kelp. So basically we have, with this different band that you can use, you can see kelp about twice as deep into the water column as you can with the near infrared. And so that was interesting in its own right, but we also wanted to look at the bed level. And so to do that, myself and Luba Rashitnik from Hakai went up and we sat on a bunch of different kelp beds on Calvert Island, and we actually took photos across the tidal cycle for a whole bunch of different beds and actually measured current at the same time. And we took those and we classified them, made orthomosaics, all that good stuff to get surface area basically at each tidal height at about 0.5 meter intervals. And basically what this showed us was that at low current sites, which is less than 10 centimeters in some of these areas, the kelp canopy decreased by about 20 to 26%, and that was regardless of species. So we had macrocystis beds, we had some nereocystis, and a mixed bed as well. But then in high current areas, so basically above 10 centimeters up to 20, which isn't even that high, we were seeing that there was almost twice as much, at some places, a decrease in kelp. So getting up to almost 39% per meter of tidal height increase. So basically, out of all of this together, we found that essentially you can detect kelp deeper with red edge versus near infrared bands. In total, with our drone imagery, we found that there's about 18% more kelp overall. That's with a high resolution drone, not necessarily with satellite imagery. But you need to be careful in shallow water because with penetrating into the water, you can also detect understory. And then in low current areas, tides are generally the dominant factor in kelp submersion and how much kelp you can see at the surface. But in the higher current areas, basically both seem to play a role. And we had sort of a low sample size for that. So there's more research needed for sure on the high current areas, especially in places like the channels around Puget Sound and southern Vancouver Island. And so I'm going to shift gears completely here and just talk a little bit about some of the ongoing work. Now I'm doing my PhD with Julia Baum in the biology department here at UVic. And so I'm going to talk about a project that we have on the go, basically looking at kelp forests in the northern Salish Sea and how that habitat has changed over the last 50 years. So this is in close collaboration with the Kelp Rescue Initiative and the Hakai Institute, and we've recently gotten some funding from Trebekage Initiative and National Geographic, as well as the Government of Canada. So some exciting stuff on the go here. And just a very quick primer, the Kelp Rescue Initiative is a new group, basically started back in 2021, but there's a whole lot on the go that we have. And generally the goal is to use science to accelerate restoration of coastal ecosystems, so kelp forest restoration, but also to better understand things like genetics and thermal tolerance and historical ecology. So there's a whole bunch of different facets to it. And all of this is largely meant to be supporting Indigenous stewardship here in BC. So we're working with a lot of First Nations along the coast, and essentially are trying to build sort of this roadmap so that nations who want to do or work towards restoration have the best tools available to do that without having to do a bunch of little case studies all over the place. And there's a whole bunch of collaborators within Kelp Rescue Initiative. I won't go into it, but we do all sorts of things, and you'll be hearing from Matt, I believe, right after me, talking about some of his work with species distribution modeling. So basically, this project in the North Salish Sea is in this area where over the past 50 or so years, sea surface temperatures in the summer have increased by about 2 degrees Celsius on average, but that doesn't really show the extreme swing. So we very constantly will have an average, recently in recent years, an average sea surface temperature of almost 20 degrees. And you get these weeks where it's like up in the mid-20s for weeks at a time. So very, very hot. Based on the last talks we just heard, you can imagine how that's going to be terrible for kelp. And there's this professor here at UVic from back in the 60s and 70s, and he went out and actually did all of this big mapping project of looking for red algae actually along the northern shores of the Salish Sea, thinking that that would be useful for industrial purposes. And so we are basically redoing these surveys. And so there's about 90 kilometers of shoreline that's mapped out, and it has these like large scale vegetation units of things like bull kelp and eelgrass and sargassum from 50 years ago. But then they also did these really good ground truthing transects. So there's about 22 ground truth transects, 219 quadrats, where they actually documented roughly 100 different species of mostly seaweeds, but also some invertebrates and things like that as well. And so this summer, we are going to be going out and resurveying this. So we've partnered with Hakai, and they're going to fly their airborne coastal observatory across that whole 90-kilometer stretch. And then we're going to be going out and doing a whole bunch of dive surveys and intertidal surveys to look at the more local scale and see how these the distribution and diversity of these species has changed.
Quick time,
and so
just a couple minutes left.
Perfect, thanks. And so basically this gives us the opportunity to look at these two scales, one being that sort of landscape scale across the whole northern Salish Sea, where we have this sort of gradient of temperature changes, and look at sort of the main habitat forming species. And then the other is looking at a more local scale and getting down to the nitty-gritty details of like what exactly, aside from just the main things like bull kelp, has actually changed in these areas. And this is just a cool photo showing some of the massive bull kelp beds we used to have. So this is up in Comox. This is the north side of Denman Island in the Strait of Georgia. And you can see this bull kelp bed is like, at least 500 meters out from shore. It continues multiple kilometers along the coastline. And these are basically all gone now and have been for quite some time now. So huge changes, and it'll be really interesting to see the associated loss of biodiversity that we can determine from these areas of really high change. Thank you. And that's all I got.
Okay, I do think we have time for 1 or 2 questions, quick ones. Any questions for Brian? Okay. Then I guess we will turn it over to Matt, also with the University of Victoria, using distribution models to evaluate the extent, composition, and trajectory of kelp forests in British Columbia.
Okay, awesome. Can you hear me?
Yep, sound good.
Awesome. So I will jump right into my presentation. Thanks for having me here to talk. Looking forward to hearing what you all think about my presentation. My name's Matt. I'm a PhD student in Julia Baum's lab at the University of Victoria. My pronouns are he, him. And yeah, I'm using species distribution models to look at the extent and composition of our kelp forests in British Columbia. So just a little bit of background to start with. Kelp forests dominate much of the temperate nearshore, occurring along an estimated 1/4 of the world's coastlines. And they are currently considered the most vulnerable temperate marine ecosystem to the impacts of climate change, with marine heat waves being identified as the largest threat to kelp on a global scale. This map shows the rough distribution of the world's main kelp genera, highlighting the large extent of kelp forests and the extreme variability in the diversity of species that make them up. In the waters that they do dominate, kelp provide important ecosystem services. They create complex three-dimensional habitat that is used by many other ecologically, culturally, and commercially important species, and increase nearshore biodiversity through increasing available niche space. Kelp-associated biodiversity has been shown to be associated with the structure made available by different kelp species and morphologies, as well as the overall structure of kelp forest environments themselves. Through its rapid growth and fixation of carbon into the food web, kelp also fuels nearshore productivity and may contribute to the sequestration of atmospheric carbon in the deep ocean. Overall, a recent review gave the global value of kelp ecosystem services provided through fisheries production, nutrient cycling, and carbon removal to be around 500 billion per year, just as a number to throw out there. So seeing as kelp forests are so valuable, it's important to understand their current trajectories. And a region that we're all very familiar with, where there is large variability in the response of kelp forest ecosystems to a variety of stressors, is the Northeast Pacific, especially British Columbia, where I'm focused, where our complex coastlines create quickly varying environmental conditions. And this part of the world has also been suggested as a potential evolutionary origin of kelp. And if you remember from the first global map I showed, has the second highest diversity of kelp species worldwide. And this makes it an excellent spot to study not only what will influence kelp forest redistributions into the future, but also what will drive community composition of these habitat forming species. And this is important to understand as we saw species identity and morphology may play a large role in the biodiversity associated ecosystem services provided by our kelp forests. And so this map is from a recent synthesis that I was a part of, which depicts the variable trajectories of kelp in BC at 11 different focal regions. And like many other studies from this area, attributes this largely to differences in temperatures experienced and trophic interactions in these regions. And a variety of modulating local factors are also postulated about. So an important caveat to note about the study that I just showed you is that it focused solely on our canopy forming species of kelp, so bull kelp and giant kelp. And these species are very important to our kelp forests in the Northeast Pacific and are easy to study thanks to the data that's available from satellites, aerial imagery, and surface boat surveys. As a result, it is common to study them when examining long-term trends and trajectories of kelp forests in the Northeast Pacific at large spatial scales. However, this focus runs a bit contrary to my previous statement that the high diversity of kelp species we have in this part of the world is likely important to the ecosystem services that these underwater forests provide us. And in fact, there are many areas of the world where kelp ecosystems don't contain these canopy forming species and yet are still highly productive in biodiversity. So as such, the methodologies I'm going to use in my thesis will focus on getting underwater and mapping kelp ecosystems as diverse communities of interacting foundation species. And the short objective statement for my thesis is basically, in general, to advance our understanding of how kelp species and communities have changed and will redistribute as environmental conditions and biotic interactions are altered by climate change, And I'll do this using data collected from underwater diver and ROV surveys, alongside continuous predictor layers to draw conclusions about important factors influencing patterns of kelp distribution and diversity in a species distribution modeling framework. So species distribution modeling is a complex undertaking, but don't worry, I'm not going to walk you through this full figure. This is a general methodological figure that I used in my recent candidacy exam. And in its simplest sense, species distribution modeling is a way of associating species records with predictor variables to develop spatial statistical models that can predict species distributions over a continuous study region using the developed relationships. And for now, suffice it to say, this breaks down into the overarching themes of data preparation, so that model fitting can occur on unbiased representative data. And a lot of model testing and validation can be done to ensure that all the distribution predictions and interpretation of predictor importance can be done in a meaningful way on these models. So doing a quick review of species distribution models for kelp on Web of Science, I found that 43 papers currently directly contain laminarian species distribution models for laminarian kelp species. And of these, 4 were global models that included BC in their extent, but did not focus on it or the questions I'm asking in the thesis at the scales I'm asking them on. And the final 2 are regional models within BC that focused on singular or a couple kelp species, Paragophora californica and our canopy forming. And this sort of highlights an important information gap in the understanding of how many of our kelp species are distributed throughout BC. My work will look to model the distribution of all common kelp species in BC at finer scales, at least 500 meters, if not finer in some situations. And different chapters I'm going to be doing are going to focus on different scales and regions. But the goal is overarchingly to create BC-wide estimates of the distribution of our kelp species and richness. And to do so, I will require 2 things, which are sort of represented on the map here: kelp occurrence data for all kelp species distributed across the large geographical and environmental gradients that we have present in BC, as well as continuous predictor layers representing all of the variables that could be important to kelp species occurrence in this part of the world. Here, I would also like to plug that I'm currently working on a chapter focused on the Salish Sea region. And while I have lots of amazing data for the Canadian side of things, I would love to talk with potential collaborators about incorporating data from Puget Sound and the other side of the Strait of Juan de Fuca to my models. And I heard some great stuff about understory kelp in Puget Sound earlier in Tim's talk. So excited to talk about with some of you here about that into the future. But with my shameless plug out of the way, let's first consider what predictor variables we could include in these models. And it's important to consider the ecological or physiological importance of each, and therefore how it could impact kelp occurrence. So in general, firstly, kelp species distributions are known to be constrained by a variety of environmental factors, and correlating these with kelp species occurrence has largely been the focus of species distribution modeling in kelp to date. Kelp are understood to mainly occur on hard rocky substrates, except in calm areas where they won't be swept away growing on little pebbles and otherwise sandy substrate. Temperature is known to be a large driver of kelp occurrence, with prolonged high temperatures leading to bleaching, tissue degradation, and death, as well as failure to reproduce and recruit. Marine heat waves are now recognized globally as the largest threat to kelp species. But temperature can be modulated by a variety of other factors, with wave energy and currents ameliorating high temperatures through mixing. And sufficient nitrate availability or nutrient availability is associated sometimes with better growth. And some studies show that this can have effects at higher temperatures. Some that we saw earlier show that doesn't necessarily. Light availability also has a direct influence on photosynthesis and growth. And hyposaline environments can lead to reduced growth, blistering, and other stress responses in kelp. In summary, there are many environmental factors influencing the occurrence of kelp, and they can interact in many complex ways, well more than I have discussed here. But in addition to these environmental drivers of kelp occurrence, we should consider the biotic interactions that mediate it. So canopy formers are known to dramatically reduce light availability and change flow and sedimentation regimes in the understory, driving differences in community composition and diversity of understory species. And so differences in the environment and availability of species, so the species pool, can also lead to differences in fitness and competition between algal species for space. So there's that interalgal dynamic there that we can think about. And then, of course, in BC, top-down trophic control has been known to sort of control that transition from algal-dominated to urchin-dominated states for some time. And with the two main urchin predators in BC, we've got sea otters and we've got sunflower sea stars, or Pycnopodia. Sea otter populations in BC were extirpated by the fur trade long ago and have since been reintroduced in certain areas of northern Vancouver Island and spread to the north and central coast. And Pycnopodia were present in large numbers throughout much of BC until populations were decimated by sea star wasting disease, becoming functionally extinct in many locations. And the loss of both of these urchin predators in many regions of BC has allowed urchins to recruit in large numbers and decimate kelp forests, making trophic controls very important to capture when modeling kelp species occurrence here, as otherwise suitable kelp habitat could now be urchin barren because of these dynamics. So all the processes that I've got represented on this slide here are going to be brought together to create the most realistic estimates of kelp distributions on a BC-wide scale. And this will be possible thanks to an amazing team of researchers who are developing fine-scale environmental layers from regional climate and ocean models and urchin density species distribution models that, while currently unavailable for all of BC, exist for some regions, such as the Salish Sea, and will be ready soon for the rest of the province. These layers will also be projected into the future so that we can postulate on how kelp extent and structure of kelp forests may change. So this is going to be done using the newest set of climate scenarios developed for the IPCC AR6 report, the Shared Socioeconomic Pathways. And each SSP represents a socioeconomic storyline that starts in 2015 and assumes different levels of mitigation and adaptation challenge, influenced by a variety of other variables and assumptions. And so recent trajectories suggest that SSP2 4.5 is the most likely future scenario in terms of its greenhouse gas emissions. And we have chosen it and SSP3 7.0 as our two future projection scenarios to capture a range of potential future warming, considering likely and less ideal future situations. And this will allow us to project kelp extent into the future. So then the final piece of this puzzle is kelp occurrence data and being able to obtain occurrence records for all of BC. And to tackle this objective, I'm working with a variety of amazing partners, including the Kelp Rescue Initiative, which Brian already introduced, Blue Carbon Canada, a project run by Julia, my supervisor, looking to assess the extent and blue carbon habitats in Canada's nearshore oceans, and then BC Parks and DFO, who have funded much of my field work or graciously provided me with their own occurrence records for my use in this modeling exercise, and many amazing First Nations who have helped carry out surveys within their traditional territories, who I would like to specifically thank now. This includes the Nampis, Kwikwisutnaqwamish, Mamalelakula, Kukachekwasit, Heshkuyat, Ahousit, Tliokuyat, Huayet, Cheatit, and Liapsit First Nations, who all supported the work in a variety of ways. So these surveys that I've been conducting with the nations were funded by BC Parks to address a lack of knowledge as to what was occurring in the benthic environments in and around many BC parks. And I used underwater ROV, or remote operated vehicle surveys, for this. Our ROV is shown in the bottom image, being held by our lab manager, Christina, and allowed for surveys to be completed much faster than with a diver and in many more locations. And this has allowed me to fill data gaps present in the benthic habitat surveys that were provided to me by DFO. For each of our ROV transects, we would drop the ROV in 15 to 20 meters of water, and we would then pilot it into the shore, taking video the entire time, trying to get as close to any of the kelp we saw as possible, so that when going back through the video later, we could ID it and associate it with a substrate class, depth, and location to use when training the species distribution models. And to do this, we used the software Beagle, which I highly recommend if you are processing this kind of data. It lets you add point and multi-frame annotations to videos and exports CSV summaries for the entire project that are easily workable in R. And this summer, I'll be heading out for more surveys, and thanks to some additional funding received, I'll be kitting up our ROV with a similar setup to that being used by Zach out of the Seattle Aquarium, who presented to this group a little while ago about their project. So that's very exciting. We'll be able to collect some percent cover metrics, hopefully, with that rather than just occurrence.
Just want to give you a quick time check, Matt.
Awesome, thank you. And so in total, we have now completed over 400 transects in 10 focal regions. And as mentioned, I'll be heading out to 4 more this summer to hopefully close to double the amount of data we have. And overall, this project is providing BC Parks with information in and around potentially 60 different protected areas. It's not been an easy undertaking, but it's beginning to pay off in the form of our first results, which I'll share now. So this map is based on 18 preliminary species distribution models that were made to show in my sort of candidacy exam using existing coarser scale environmental data for all of BC, so not the models that I'll be using in my final pass, but still require a lot of refinement as shown by the large bed of kelp predicted off the side of Haida Gwaii, which definitely doesn't occur there. And so I would ask everyone to take these patterns with a grain of salt. But with that said, what is shown here is the kelp richness throughout the province. And if we zoom in on a couple areas like the Strait of Juan de Fuca and the God's Pocket region at the top of Vancouver Island, we can see that high richness is associated with these high current, high exchange areas, as we might expect, as this increases mixing, provides nutrients, cools things down for kelp. And if we look to the north of BC, we're also seeing just generally higher richness there. As well, we have the Salish Sea here, where we see, especially in the north, so in the Strait of Georgia, very low kelp richness predicted, which matches with trends we see in our on the ground surveys. In addition, bringing this back to the canopy forming versus understory focus, initial pass of our models, so those 18 models again, showed that canopy formers are actually found in less than 40% of our predicted kelp ecosystems in BC. So interesting to think about when we're thinking about the interactions between these species and what's found in the understory for sure. And so with that, I would like to say thanks to my team who's made this all possible, and thank you all for listening. And I can take questions, and I've got my email and contact information up here so that you can ask questions further as well.
Elizabeth, you're muted.
Oh, yeah, I think you're muted, Elizabeth.
Elizabeth, I'm going to jump in because I see you're muted. Thank you so much, Matt. We'll go ahead and move you, the discussion following your talk to the chat. And I just started with a first question for you. Great talk. And now, so it's 11:50. It's 11:52 actually. You have been listening to this for almost 2 hours. So first, what we'd like to do is thank all of these speakers. It's just been an amazing agenda, and it makes, I think, us all feel so much better that we're learning so much. So thank you, everyone. And then, are there any last issues? We have about 5 minutes left that someone would like to bring up, or shall we just have 5 extra minutes in our lives? So anyone who wants to could raise their hand, or and if I don't see, I see lots of nice applause, but if there aren't any issues, I'd say let's call it a wrap.
I will say I'll keep the meeting open so that people can continue to use the chat until the top of the hour, since there are some questions coming in over chat, if that's okay. So you can stick
sure.
around
And then
rest is free to go.
Yes. Thank you, everyone else. And the last thing I would say is we always are looking to make sure we hear from people who are doing interesting research that should be shared. So send us an email about something you're doing or something you know that's happening that you think would be really good to hear about in this group. And then the one other concept is today we did a rapid series of talks. We can, we do different formats every time we have one of these meetings. So if you have a particular format that you would like, let us know about that too. So thank you all so much. It's great that you're all a part of this work group. We really appreciate you.
Thanks, Helen.
Thanks, Helen and Elizabeth, for hosting.
You're welcome.
Yeah, and for all the speakers that are still on, I hope you're looking at the chats Everyone loved it. It's such an honor to look at this lineup.
I'm still tagging my my answers as well.
I was gonna say, I can keep it open. Feel free for those who want to step away, but I'll keep the chat open for those who want to respond to some questions, and I can put it in the meeting summary.
Helen, do you know if—
Hey, Jenny.
Hi, good to see you. What an amazing team of seaweed knowledge keepers we are evolving in this area. It's just really exciting. I would love to know if you know about Nereocystis luetkiana and if it might be listed and who's working on that. You know, listed for—
Oh yeah, you know, the person who's right on that is I think of most as Dave Lowry. And he gave us an update at the last work group meeting. So you could look on the website. I can, I don't know if Elizabeth, if you could add that link to the chat. And then it looks like Hillary's going to add something. What do you got, Hillary?
I was just typing out an answer to you there, Jenny.
Oh, Hillary, thank you.
Yeah, just from what I understand, and this is coming from Dave, and then also I talked to somebody who's somebody else in NOAA who's working specifically on the Nereocystis thing. This petition was filed last fall, and they're a little behind in deciding how far they're going to take it, but presumably they'll have to do lots of rounds of considerations if they decide. Dave describes it as the bar is pretty low for having enough evidence submitted that it has to have a full consideration. So that's most likely, and that I've heard that hasn't gone through yet, although it could just be behind on information. And then that would be likely a process of multiple years. One of the complex things about Nereocystis is that we don't really understand the subpopulations. Like it's all just kind of one meta population throughout the range. So that means that unless that knowledge is better developed, as I understand it, it would just be like listed for the whole West Coast. And so that's something that obviously we'd be concerned about. And maybe the emerging genetic information can parse out some of that in the years of the consideration of whether it would be listed as threatened or not. And then there could be specific recommendations in certain areas. So that's a lot of hearsay, but we've been talking about this with some NOAA folks for sure.
Okay, that's very, very helpful. Thank you. And do you know, is someone doing the genetic evaluation now?
There's a couple different projects. So we have one with Felipe Alberto at University of Wisconsin that's really focused on Washington-based Nereocystis, and then Kelp Rescue and some of their collaborators in BC have another one that is got some. I think they've got more outgroups from along the coast, but I'm not positive exactly what is being included there.
Hillary, very helpful. Thank you. I was in the dark on that one.
Yeah, and thanks for sharing today. It was great to hear you talk about your work. We've been referencing that paper, so very interesting.
Well, my pleasure. I stand on the shoulders of many. It's a big team, as we all know. None of us do this single-handedly. We have wonderful assistants, and you all are extremely inspiring. So, all right. Well, I got to go back to my teaching mode. 2 more weeks. Take care, everybody.
Good luck.
Good to see new faces.
Thanks, Helen and Elizabeth. Thanks, everybody.
Yes.
Thanks, everyone. It's good. So good to see you all.
And I'm gonna close it down because the chat looks quiet. So I'll see you later, Helen. Enjoy the Outer Banks.
Take care. And hi, Megan and Zach. I didn't get a chance to say hi to you yet. Bye.
Good to see you all. Take care.